श्री तुम तो ऐसे बोल रहे हो कि जैसे मैं शुभम को पर्सनली जानती हूँ। मुझे कैसे पता होगा वो ज्वाइन करेगा कि नहीं करेगा। Just assumption he is dedicated. I don't think उसे लगा होगा ना मैं झूठ बोली, मैंने बोला था कि मैं 3 पे आऊंगी लेकिन मैं 3 पे नहीं आई, तो आई थिंक ही स्लेप्ट। वैसे मेरे पास काफ़ी सारे लोगों का कांटेक्ट था, एलेक्स, शुभम, निधि, सर्वजीता, भवा, सबका था, लेकिन हम लोगों ने खो दिया। हाय आकांक्षा। गुड मॉर्निंग टू यू एज वेल। मुझे ना आज भी मेरे पास टाइमर नहीं है ना, तो थोड़ा अजीब लगेगा कि विदाउट टाइमर पढ़ने में, क्योंकि सबसे पहले जो करते थे हम लोग टाइमर ही सेट करते थे। दैट्स काइंड ऑफ वियर्ड। टेलीग्राम, हाँ वो टेलीग्राम डीएक्टिवेट नहीं, डिलीट ही कर दिया। मैं वापस टेलीग्राम पे नहीं आना चाहती, वरना हम लोगों को एक ग्रुप था टेलीग्राम पे, यूट्यूब वाले ही फ्रेंड थे, ज़्यादा नहीं थे, आई थिंक 8 समथिंग थे। यानी को छोड़ के, यानी को मैं पर्सनली जानती हूँ। तो पढ़े स्लॉट 1 रहेगा ना ये हम लोगों का, 90 मिनट का डायरेक्ट करते हैं, ओके 1.5 घंटा का, क्योंकि सुबह सुबह ना ज़्यादा मन करता है पढ़ने का। फर्स्ट सेशन ज़्यादा लंबी देर तक खींच सकता है ना। स्लॉट 1 90 मिनट, 4 5 पे स्टार्ट किया तो 5:30, 6:30 पे ब्रेक लेंगे। फाइव मिनट ब्रेक अभी नहीं, बाद में लेंगे। एक स्लॉट कंप्लीट करने के बाद 5 मिनट का हम ब्रेक ले लेंगे। अब गुड लक, हाँ कोई भागना मत, आकांक्षा भागना मत, श्री पढ़ना, ठीक है, तुम हमेशा भाग जाते हो बीच में। श्री I think Yuvraj is there. Are you there, Yuvraj? Okay, Akanksha. Good luck everyone. Good morning, Nova. अब मैं चार्ट्स नहीं देख सकती, ओके मैंने क्लोज कर दिया। मैं खुद के लिए यहाँ से भी चार्ट्स हाइड कर देती हूँ। डन, मैंने हाइड कर दिया खुद के लिए। गुड लक युवराज। यहाँ से भी हाइड कर देती हूँ, 1 मिनट को। हाँ कर दिया। माइक्रोपाइलर एंड न्यू सर्विस। मैंने ब्रेक कब बोला था? ब्रेक एट 6:30। तो ये तो 90 मिनट का नहीं, 100 मिनट का हुआ, सेंचुरी का हुआ। 25 पे ले लेंगे, कोई बात नहीं, ठीक है। Micropylar end, new cellus, megaspore, mother cell, megaspore, mother cell, new cellus, micropylar end, new cellus, megaspore dyad, megaspore tetrad, micropylar end, megaspore tetrad, megaspore dyad, new cellus. 2, 4 and 8 nucleate. 2, 4 and 8 nucleate. Okay. 7 celled, 8 nucleate. Synergids 2, egg cell 1. 2 synergids and 1 egg cell. 1 central cell, 2 polar nuclei. Antipodal. Chalaza and antipodal. This is orthotropous. Okay. Micropylar end. Filiform apparatus. Synergids. Okay. Chalaza and antipodal. Chalaza and antipodals. Polar nuclei. Central cell. Egg. Synergids. Filiform apparatus. Micropylar end. The egg apparatus in turn consists of synergid and 1 egg cell. The synergid have a specialized cellular thickening at the micropylar tip called filiform apparatus. They are called filiform apparatus, which plays an important role in guiding pollen tube into the synergid. 3 cells at the chalaza end and are called antipodal. The large central cell, as mentioned earlier, has 2 polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8 nucleate, is 7 celled. Okay. The nucleus of the functional megaspore divides mitotically to form 2. ये तो मैंने किया था, लेकिन फिर भी एक बार कर लेती हूँ. The nucleus of the functional megaspore divide mitotically to form 2 nuclei, which moves to the opposite pole. The nucleus of the functional megaspore divides mitotically. The nucleus of the functional megaspore divide mitotically to form 2 nuclei, which move to the opposite pole, forming the 2 nucleate embryo. ये तो हो गया था ना. 2 nucleate, फिर 4 nucleate, फिर 8 nucleate. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. Except the distribution of cells. Polar nuclei situated below the egg apparatus in the large central cell. Central cell, egg, nucleus. Okay, these are synergids. Plasma membrane, filiform apparatus, male gamete, vegetative nucleus. Pollen tube, antipodal cell, polar nuclei, egg cell and synergids. Pollen tube. Those are stigma. True chemi. Longitudinal section of a flower showing growth of pollen tube. Antipodal cell, pollen tube, synergid, egg cell, then polar nuclei, finally antipodals. Orthotropous. I think I'll die. Pollen grain germinating on the stigma. Pollen tube growing through the style. lower section, longitudinal section of pistil, showing path of pollen tube growth. Enlarged view of an egg apparatus showing entry of pollen tube into a synergid. Discharge of male gamete into the synergids and the movement of the sperm, one into the egg and the other into the central cell. One into the egg and the other into the central cell. A vegetative and a generative cell. In such cell, the generative cell divides and forms the two male gametes during the growth of pollen tube in the stigma. In plant which shed pollen in the 3-cell condition, pollen tube carry the two male gametes from the beginning. Stigma style, ovary and thalamus. This is syncarpus, poppy plant, papaver, syncarpus. This is Michelia apocarpus. Hilum is a junction between funicle and ovule. Funicle is the stalk that attaches ovule to placenta. Hilum, then micropylar pole, outer integument, testa, inner integument, tegmen, nucellus, and finally embryo sac. This is anatropous structure, okay. Vacuoles nucleus, asymmetric spindle, vegetative cell, stores abundant reserve food material, and they have prominent nucleus, not prominent, they have irregular shaped nucleus. Am I right? They are bigger, okay. The vegetative cell is bigger, has abundant food reserve and large irregularly shaped nucleus, okay. Small fluids in the cytoplasm of vegetative cell. It is a spindle shaped with the dense cytoplasm and a nucleus. Dense cytoplasm and nucleus because it undergoes division to form two male gamete. Parthenium. Asymmetric spindle. Vacuole nucleus, asymmetric spindle. Vegetative cell, okay. Generative cell. Scanning electron micrographs of a few pollen grains. Epidermis, endodermis, middle layer, microspore mother cell. This one is epidermis. Then we have endodermis, endodermis, middle layer, and finally microspore mother cell. Dictum. Epidermis, endodermis, middle layer, microspore mother cells. Dictum. Connector, epidermis, okay. Connector, epidermis, endothecium, sporogenous tissue. Dictum, middle layer, endodermis, epidermis, okay. There is connector too. अब मैं डबल फर्टिलाइजेशन स्टार्ट करूँगी. आई थिंक यहाँ से मुझे थोड़ा टफ भी लगेगा. Excuse me. ह्यूमन रिप्रो, उसके बाद है ना क्या है, प्रिंसिपल ऑफ इनहेरिटेंस कर लेंगे, एवोल्यूशन रहेगा। एवोल्यूशन आई थिंक तुम्हें इसके बाद एवोल्यूशन ही करना चाहिए, तो चैप्टर चला जाएगा, तो दो ही बचेगा ना। स्ट्रक्चरल ऑर्गेनाइजेशन भी तो बचा है, आई थिंक सिलेबस तो कवर होगा पाएगा। तो double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. Endosperm nucleus in endosperm. Nucleus is 3N. Okay. After entering one of the synergid, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. Okay. Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of diploid cell, the zygote. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, pen. Double fertilization. After entering one of the synergid, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gamete moves towards the egg and fuses with its nucleus, thus completing the syngamy. Fusion of egg and sperm, male gamete. Syngamy. This results in the formation of diploid cell, the zygote. Zygote is a diploid cell. The other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, pen. Primary endosperm nucleus, triploid. As this involves the fusion of three haploid nuclei, it is termed triple fusion. Three haploid nuclei. Okay, fusion of three haploid nuclei is termed as triple fusion. Fusion of three haploid nuclei. Since two types of fusions, syngamy and triple fusion, takes place in the embryo sac, the phenomena is termed double fertilization. Since two types of fusions, syngamy and triple fusion takes place in an embryo sac, the phenomena is termed double fertilization. The phenomena is termed double fertilization. An event unique to flowering plants. Since two types of fusions, syngamy and triple fusion, takes place in an embryo sac. Since two types of fusions, syngamy and triple fusion, takes place in an embryo sac, the phenomena is termed double fertilization, an event unique to flowering plants. एम्ब्रियो साकभित्र दुईटा फ्युजन हुँदैछ, एउटा त हाम्रो सिनग्यामी हो, त्यहाँदेखि अर्को चाहिँ के हो, सिनग्यामी एन्ड ट्रिपल फ्युजन. एउटा सिनग्यामी हो, अर्को के हो, ट्रिपल फ्युजन. Since two types of fusions, syngamy and triple fusion, takes place in an embryo sac, the phenomena is termed as double fertilization, an event unique to flowering plants. दुईटा टाइपको फ्युजन अरे two types of fusions in gamete and triple fusion takes place in embryo sac. The phenomena is termed double fertilization, event unique to flowering plants. The central cell part. The central cell after triple fusion becomes the primary endosperm peck. Double fertilization, fusion gamete and triple fusion. Okay. The central cell after triple fusion becomes the primary endosperm cell. Triple fusion के बादमा primary endosperm cell बनिन्छ and develops into the endosperm, while the zygote develops into an embryo. Zygote develops into an embryo. Peck, primary endosperm cell develops into an endosperm. Primary endosperm cell. Peck develops into endosperm, whereas zygote develops into embryo. Primary endosperm cell. Fertilized embryo sac showing zygote and primary endosperm nucleus pen. Degenerating synergids. Zygote to an primary endosperm cell peck. Primary endosperm nucleus. The ploidy of pen is 3n. Ploidy of pen is 3n. Degenerating antipodal cells. Degenerating synergids. Degenerating antipodal cell 3. Degenerating synergids 2. Zygote. Ploidy of zygote is 2n. Primary endosperm cell. Primary endosperm nucleus 3n. Pen. Ploidy of pen is 3n. Pen. Primary endosperm nucleus develops into endosperm. And zygote develops into embryo. Zygote. Globular embryo. Heart-shaped embryo. Then finally, mature embryo. Heart-shaped embryo, mature. There is plumule, cotyledons, radicals, and suspensor. Radical, suspensor, heart-shaped embryo, plumule, mature cotyledon. Okay. Post-fertilization, structure and event. I think it's fine, isn't it? Following double fertilization, events of endosperm and embryo development. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruits are collectively termed post-fertilization events. Post-fertilization structure and events. Following double fertilization, event of endosperm and embryo development, maturation of ovule into seed and ovary into fruits are collectively termed post-fertilization events. Fertilized embryo sac, zygote, showing zygote and primary endosperm pen, stays in embryo development in a dicot. Okay. Fertilized embryo sac showing zygote and primary endosperm nucleus, I think. Following double fertilization event of endosperm and embryo development. Event of endosperm and embryo development. Endosperm development and embryo development. Maturation of ovule into seed and ovary into fruit. Maturation of ovule into seed. Ovule matures into seed and ovary into fruit are collectively termed post-fertilization event. Maturation of ovule into seed and ovary into fruit are collectively termed as post-fertilization event. Post-fertilization. Endosperm and embryo development. These are post-fertilization. Okay, development of endosperm and embryo. Then formation of seed, formation of fruit. These are all post-fertilization event. Maturation of ovule into seed. Ovule seed ma baninu, ovary fruit ma baninu. Post-fertilization event. Okay, I think I can. Following double fertilization events of endosperm and embryo development, maturation of ovule develop into seed and ovary into fruit. Okay, are collectively termed post-fertilization event. Following double fertilization events of endosperm and embryo development, maturation of ovule into seed and ovary into fruits are collectively termed post-fertilization events. Endosperm. Endosperm development precedes embryo development. Endosperm development precedes embryo development. Why the primary endosperm cell divides repeatedly and form a triploid endosperm tissue? Primary endosperm development precedes before embryo development. The primary endosperm cell divides repeatedly and forms a triploid endosperm development precedes embryo development. Okay, endosperm development precedes embryo development. Why the primary endosperm cell? Okay, the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. Triploid endosperm tissue. The cells of this tissue are filled with reserved food materials and are used for the nutrition of the developing embryo. The primary endosperm cell divides repeatedly and forms a triploid. The primary endosperm cell divides repeatedly and forms a triploid. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. Triploid endosperm tissue. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. triplets, repeatedly and forms a triplet endosperm tissue. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissues are filled with reserve food material. Endosperm tissue, primary, a triploid endosperm tissue. Triploid endosperm tissue. The primary endosperm cell divides repeatedly and form a triploid endosperm tissue. Triploid endosperm tissue. Excuse me. The cells of these tissues are filled with reserve food material. The cells of these tissues are filled with reserve food material and are used for the nutrition of developing embryo reserve food material. Triploid endosperm tissue, okay. In most common type of endosperm development, pen that is primary endosperm primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. In most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. This stage of endosperm development is called free nuclear endosperm. In the most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, and bean before seed maturation, or it may persist in the mature seed, example castor and coconut, and consumed and used up during seed germination. Split open some seed of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of these tissues are filled with reserve food materials and are used for the nutrition of the developing embryo. In most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. Endosperm tissue. वो बबल उड़ाने में मज़ा आता है ना, क्लिक करके इमोजी फेंकने में, मुझे भी मज़ा आता है। मैं भी ऐसा ही करती हूँ दूसरों के लाइव में जाके। पढ़ाई करो अभी तुम। Why the primary endosperm cell divides repeatedly and form a triploid endosperm tissue? The cell of this tissues, triploid endosperm tissue. Triploid, मुझे तो भूख लग रहा है, मुझे कुछ खाने का दिल कर रहा है। Triploid endosperm tissue. Why the primary endosperm cell divides repeatedly and form a triploid endosperm tissue? The cells of these tissues are filled with reserve food material and are used for the nutrition of developing embryo. Developing embryo को लगी food जस्तो के हुन्छ used हुन्छ. The cells of these tissues are filled with reserve food material. Endosperm. In the most common type of endosperm development, the pin undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm. No, I'm very happy with this. Like, I think 2019, right? 1920? No, before that, 2018 or something like that. Mm-hmm. Stain. Where was I? Primary endosperm nucleus undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but the free nuclear endosperm, made up of thousands of nuclei. And the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo. Example, pea bean and the groundnut. Pea, groundnut and beans, okay? Before seed maturation, or it may persist in the mature seed. Example, castor and coconut may persist in the mature seed. Example, castor and coconut, and utilized while germinating seed can be and be used up during seed germination. Castor and coconut, they are endosperm, okay? Castor and coconut, persistent endosperm, and be used up during seed germination. Split open some seed of castor, peas, beans and groundnut. Split open some seed of castor, peas, beans and groundnut. भूख लगेगा अम्बू
पकाएर खायो भने त मलाई लाउनु पर्यो
किन खुद्दैन यहाँनिर आएको Endosperm may either be completely consumed by the developing embryo. Example, pea, groundnut, bean. Before cell, मैं अम्मा को बोल रही हूँ कि मुझे भूख लगा है. अम्मा बोल रही है कि पका के खा ले. Endosperm may either be completely consumed by the developing embryo. Example, pea, groundnut, beans. Before seed maturation, or it may persist in the mature seed. Example, castor and coconut, and be used up during seed germination. Split open some seed of the castor, pea, bean, groundnut, fruit of coconut and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Yes, endosperm is persistent. Okay. Zygote, primary endosperm cell, like primary endosperm nucleus, pin 3N. Okay. Triploid, ploidy, zygote. Then zygote बाट के हुन्छ भन्दा heart shape अहिले नै हुँदैन, globular first हुन्छ, globular embryo, zygote, pen, globular embryo, then finally heart shaped embryo. It has suspensor. Heart shaped embryo has suspensor, and along with this, what else we have? Heart shaped embryo, suspensor. We have mature embryo, that is, it has radical, pumil, and cotyledon. Along with the suspensor, radical, pumil, and cotyledons. Okay. Heart shaped embryo. Okay, embryo finally. Embryo develops into micropylar end of the embryo sac. Embryo develops at the micropylar end of the embryo sac. Micropylar ma. Yes, this is Chalaza and Chalaza. Micropylar. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differs greatly, the early stages of different development. Though the seed differ greatly, the early stages of embryo development, embryos, early development of embryo, early stage of embryo development, embryos, early stages of embryo development. Early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. The early stages of embryo development, embryos, are similar in both monocotyledons. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differ greatly, the early stages of embryo development, embryogeny, are similarly in both monocotyledons and dicotyledons. Okay, मैंने बस एक पेज पढ़ा क्या 30 मिनट में भाई साहब. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. The zygote give rise to pro-embryo and subsequently to the globular embryo. These are pro-embryo, okay? Pro-embryo. The zygote gives rise to pro-embryo, then globular embryo, finally heart-shaped embryo and mature embryo. Globular pro-embryo, zygote pro-embryo, globular embryo and heart-shaped embryo. Finally, mature embryo. A typical dicotyledonous embryo, 2.14a. A typical dicot embryo, okay? Pumil, cotyledons, hypocotyle, radical and root cap. Radical, root cap, hypocotyle, pumil, epicotyle and cotyledons. Root cap, radical, hypocotyle. Pumil radical को PHR, RHP, PHR. Pumil, hypocotyle, radical, root cap. Pumil को माथि के छ epicotyle and cotyledons. Pumil, epicotyle, cotyledons, hypocotyle, radical and root cap. This is a typical dicot embryo. Root cap, radical, hypocotyle, pumil, cotyledons. scutellum, coleoptile, shoot apex, epiblast, radical, root cap, and coleorhiza, root cap, radical, cleoptile, coleoptile bandam bhaiya, shoot apex, epiblast, this is epiblast, and finally scutellum. Scutellum is a cotyledon. Okay, a typical dicot embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is the epicotyle, which terminates with the pumil or stem tip. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminates with the pumil or stem tip. Typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is called epicotyle. Above the level. The portion of the embryonal axis above the level of the cotyledon is called epicotyle, which terminates with the pumil. Or stem tip, pumil or stem tip. The cylindrical portion below the level of the cotyledon is hypocotyle. Below the level of the cotyledon is hypocotyle. That terminates as its lower end in the radicle or root cap. That hypocotyle that terminates at its lower end in the radicle or root cap. Okay. The cylindrical portion below the level of cotyledon is hypocotyle. The cylindrical portion below the level of cotyledons. The cylindrical portion below the level of cotyledons is hypocotyle that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. The cylindrical portion below the level of the cotyledon is hypocotyle that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. The tip is covered with a root cap. Okay. Radical, root tip. Hypocotyle below the level of cotyledon, right? Above the level of cotyledon is epicotyle. Below the level of cotyledon is hypocotyle. Embryos of monocotyledons, they have epiblast. I don't know the function of epiblast. Does anyone knows? What is the function of epiblast? Embryo of monocotyledons possesses only one cotyledon. In grass family, the cotyledon is called scutellum. Embryo of monocotyledons possesses only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryo axis. Embryonal axis. Embryos of monocotyledons possesses only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. At its lower end, the embryonal axis has. In the grass family, the cotyledon is called scutellum, that is situated towards one side lateral of the embryonal axis. Embryo of monocotyledons possesses only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side, lateral of the embryonal axis. That is situated towards one side, lateral of the embryonal axis. Embryo of monocotyledon possesses only one cotyledon. In the grass family, the cotyledon is called scutellum, that is situated towards one side, lateral of the embryonal, one side, lateral of the embryonal axis, towards one side, embryonal axis, shoot apex, radical, root cap, coleorhiza, coleoctile, shoot apex, radical, root cap or coleorhiza. At its lower end, the embryonal axis has the at its lower end, the embryonal axis has the at its lower end, the embryonal axis has the radical and root cap, enclosed in an undifferentiated sheath called coleorhiza, whatever. In this at its lower end, in the grass family, the cotyledon is scutellum, that is situated towards one side, lateral of the embryonal axis. At the lower end, the embryonal axis has the radical and root cap enclosed in undifferentiated sheath. I don't like it, but anyways, study. Radical and root cap enclosed in an undifferentiated sheath called coleorhiza. At its lower end, and the embryonal axis has the radical and root cap enclosed in the undifferentiated sheath called coleorhiza. Lower end upper end lower end. At its lower end, embryonal axis has the radical and the root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Above the level of attachment of scutellum. Above the level of the attachment of scutellum is the colectile. The portion of the embryonal axis above the level of the attachment of scutellum is the epicotyle. So epicotyle, scutellum. Epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, the colectile. That is epicotyle. Above the level of the attachment of scutellum is the epicotyle. The epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure, colectile. Soak a few seeds in water, say of wheat, maize, peas, chickpeas, groundnut overnight, then split the seed and observe the various part of the embryo and of the seed. Epiblast. Let me check. What is the major function of epiblast?
The primary function of the epiblast is to give rise to the embryo proper and its associated tissues. During early development, the epiblast, a layer of cells in the inner cell mass, differentiates into the three primary germ layers, ectoderm, mesoderm.
The primary function of epiblast is to give rise to the embryo proper and its associated tissue. To give rise to the embryo proper and its associated tissues. During early development, the epiblast, a layer of cell in the inner cell mass differentiate into three primary germ, ectoderm, mesoderm, and endoderm, and forms the foundation of the developing embryo. Okay, डेवलपमेंट ऑफ एम्ब्रियो में हेल्प करता है. ठीक है, क्या कर सकते हैं? कुछ नहीं कर सकते, बस पढ़ाई कर सकते हैं. Enclosed in a hollow folioid structure, the coleoptile. अभी भी बहुत है पढ़ने के लिए तो. Seed. In angiosperm, the seed is the final product of sexual reproduction. Seed is the final product of sexual reproduction. It is often described as the fertilized ovule. Seeds are formed inside fruit. A seed typically consists of seed coat, cotyledons, and an embryo excess. Seed coat, cotyledon, and embryo excess. The cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserve, as in legumes. Cotyledon of the embryo are simple structure, very generally thick and swollen due to the storage of food reserves, as in legumes. Mature seed may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm, as it is completely consumed during embryo development. Example: pea, groundnut, albuminous. Okay, pea and groundnut, that's it. The non-albuminous seed have no residual endosperm, as it is completely consumed during the embryo development. Example: pea, groundnut. Albuminous seed retain a part of endosperm, as it is not completely used. Albuminous seed retains a part of endosperm, as it is not completely used. Albuminous seed retains a part of endosperm, as it is not completely used up during embryo development. Example: wheat, maize, barley, castor. Albuminous seeds retain a part of endosperm, as it is not completely used up during embryo development. Example: wheat, maize, barley, castor. Wheat, maize, barley, castor. These are albuminous seed. Retain a part of endosperm, as it is not completely used up during embryo development. Okay, non-albuminous ले डल्लै use गर्छ, ex-albuminous or albuminous. Albuminous ले के गर्छ, retain गर्छ. Okay, the cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserves, as in legumes. Mature seed may be non-albuminous or ex-albuminous. Non-albuminous or ex-albuminous. Non-albuminous seed have no residual endosperm, as it is complete. Non-albuminous seed have no residual endosperm, as it is completely consumed during embryo development. Example: pea, groundnut. Non-albuminous seed have no residual endosperm, as it is completely consumed during embryo development. Example: pea, groundnut. Albuminous seed. Non-albuminous seed have no residual endosperm, as it is completely consumed during embryo development. Pea and groundnut. Part of endosperm, as it is not completely used up during embryo development. Example: wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also present. This residual persistent nucellus is called perisperm. Perisperm. Black pepper and beet. Black pepper and beet. Remnant of nucellus in some seed. Just black pepper and beet remnants of nucellus are also persistent. The residual persistent nucleus is called perisperm. Okay, perisperm. Integuments of ovules harden as tough protective seed coat. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. Entry of oxygen and water during the seed germination. Integuments of ovule harden. Integuments of ovules hardens as tough protective seed coat. Okay, integuments seed coat. Integuments. मैं फ्रूट कोट सोच रही थी दिमाग में. टफ सीड कोट है ये. सीड कोट भाई साहब. सीड कोट. सीड के लिए भी जैकेट चाहिए ना, तो सीड कोट है इंटीग्यूमेंट्स. सीड की जैकेट. Integuments of ovule harden as tough protective seed coat. Seed coats. Okay. The micropyle remain as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. I think we'll be fine. Just पढ़ना पड़ेगा. 6. Integuments of ovule harden as tough protective seed coat. The micropyle remain as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, the water content is reduced and seed becomes relatively dry, 10 to 15% moisture by mass. As the seed matures, its water content is reduced and seed becomes relatively dry, 10 to 15% moisture by mass. Moisture by mass. The general metabolic activity of the embryo slows down. General metabolic activity of embryo slows down. The embryo may enter in a state of inactivity. Embryo may enter a state of inactivity called dormancy. Or if favorable conditions are available, adequate moisture, oxygen and suitable temperature, they germinate. As ovule matures into seed, the ovary develops into fruit. That is the transformation of ovules into seed and ovary into fruit. आई मुझे वो नहीं समझ, मुझे नहीं सोचना है उसके बारे में प्लीज. मैं क्यों डिस्ट्रैक्ट हो रही हूँ? As ovule matures into seed, the ovary develops into fruit. As ovule matures into seed, the ovary develops into a fruit. That is, I hate her. As ovule matures into seed, the ovary develops into fruit. That is, the transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into the wall of fruit called pericarp. The wall of ovary okay. Ovary called cancer develops into the wall of fruit called pericarp. The fruit may be fleshy as in guava, orange, mango, etc. or may be dry as in groundnut and mustard, etc. Fleshy in guava, orange and mango. Dry as in groundnut and mustard, etc. Many fruits have evolved mechanism for dispersal of seed. Many fruits have evolved mechanism for dispersal of seed. Okay, many fruits have evolved mechanism for dispersal of seeds. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? As ovule matures into seed, the ovary develops into fruit. That is, the transformation of ovules into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. As ovule matures into seed, the ovary develops into fruit. That is, the transformation of ovule into seed and ovary into fruit proceeds proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. As ovules matures into seed, the ovary develops into the fruit. That is, the transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into the wall of fruit called pericarp. The fruit may be fleshy, as in guava, orange, and mango, or may be dry, as in groundnut and mustard. Many fruits have evolved mechanism for the dispersal of seeds. Recall the classification of fruits. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovule in an ovary and the number of seed present in a fruit? Many fruits have evolved mechanism for dispersal of seed. Recall the classification of fruits and their dispersal mechanism which you have studied in an earlier class. Recall the classification of fruit. मैं confused हो रही हूँ, but कोई बात नहीं. Recall the classification of fruits and their dispersal mechanism that you have studied in an earlier class. Is there any relationship between number of ovules in an ovary and the number of seed present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and falls off. However, in a few species such as apple, strawberry, and cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruit. Most fruit, however, develops only from the ovary and are called true fruit. In most plants, by the time the fruit develops from the ovary, other floral appendages degenerate and fall off. Apple, strawberry, cashew, etc. The thalamus also contribute to fruit formation. Okay, such fruits are called false fruit. मुझे भी ना हम लोगों को ऑनलाइन स्ट्रॉबेरी की सीड्स मिल सकती है क्या? मिल सकती है? मुझे भी स्ट्रॉबेरी को ग्रो करना है, ऐसा हो रहा है. In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruits are called false fruit. Most fruit, however, develops only from the ovary and are called true fruits. Most fruits, however, develops only from the ovary and are called true fruits. Apple, strawberry, and cashew. And are called true fruits. Although in most of the species, fruits are the result of fertilization, there are few species in which fruit develops without fertilization. Fruits are the result of fertilization. There are few species in which fruit develops without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Tomato. Parthenocarpy can be induced through the application of growth hormone, and such fruits are seedless. Okay, parthenocarpic fruits are seedless. Okay. Auxin and gibberellins. induce parthenocarpy. Growth hormone induce parthenocarpy. Auxin and gibberellin. Auxin and gibberellin induce parthenocarpy. Auxin and gibberellin induce parthenocarpy. False should be. It is true fruit. Integuments of ovule harden as a tough protective seed coat. Seed coat. Integuments. Seed coat. Cotyledon. Shoot apical meristem. Hypocotyl. Shoot apical meristem. Hypocotyl. Root tip. Endosperm. Okay. Seed coat. This is A. Structure of some seed. Structure of some seed. Cotyledon micropyle. Cotyledon micropyle. Seed coat. Cotyledon. Shoot apical meristem. Hypocotyl root axis. Hypocotyl root axis. Endosperm. Root tip. This is root tip. Hypocotyl root axis. Root tip. Hypocotyl root axis. Shoot apical meristem. Cotyledons. Those are endosperm. Seed coat. Seed coat. Cotyledons. Okay. Seed coat. Cotyledons. Shoot apical meristem. Hypocotyl and roots. Root tip. Endosperm. Okay. Cotyledon micropyle. Seed coat, and this is endosperm. This is cotyledons. Cotyledons, endosperm, seed coat. Seed coat, endosperm, cotyledons. Hypocotyl root axis. Hypocotyl root axis. Root tip. Shoot apical meristem. Shoot apical meristem. Root tip. Hypocotyl root axis. Pericarp. Endosperm. Scutellum. Scutellum. Endosperm. Pericarp. Coleoptile, pumale, radical, coleorhiza, radical, pumale, coleoptile. Thalamus, seed, endocarp, mesocarp. Thalamus, acne, acne, acne. Who cares? Banana is one such example. Tomato. Parthenocarpy can be induced through the application of growth hormone, and such roots are seedless. Okay. Seeds offer several advantages to angiosperm. Firstly, since reproductive processes such as pollination and fertilization are interdependent of water, seeds offer several advantages to angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitat and helps in species own. Okay. Seed coat, endosperm, cotyledon, hypocotyl root axis, shoot apical meristem, root cap, root tip, hypocotyl root axis, root tip, shoot apical meristem. Okay. Pericarp, endosperm, scutellum, endosperm, pericarp. Alluvium, pericarp, endosperm, scutellum, coleoptile, pumale, radical, coleorhiza. Scutellum, endosperm, pericarp. Okay. Thalamus, acne. Thalamus, fruit is developed from apple, maybe. Apple, strawberry, and cashew. Seed. Endocarp, mesocarp. Endocarp and mesocarp. Thalamus. Seed. Okay. False fruit of apple and strawberry. Seed offers several advantages to Parthenocarpy. Auxin र gibberellinले induce गर्छ. Auxin and gibberellin induce Parthenocarpy. Seed offers several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Seed offers several advantages to angiosperm. Seed offers several advantages to angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Seeds have better adaptive strategies for dispersal to new habitat and helps the species own. The hard seed coat provides protection to the young embryo. Being product of sexual reproduction, they generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage of seeds, which can be used as food throughout the year. Dehydration and dormancy of mature seeds are crucial for storage of seed, which can be used as food throughout the year and also to raise crop in the next season. Can you imagine agriculture in the absence of seed or in the presence of seed which germinate straight away soon after formation and cannot be stored? How long do the seed remains alive after they are dispersed? This period again varies greatly. Okay, pollen grain viability उसमा कम्ती थियो. Wheat and rice मा 30 minute, whereas rosaceae, leguminaceae and solanaceae मा I think 1 month जति थियो, few months जति थियो. See, rice and wheat pollen lose viability in 30 minutes. Rosaceae, leguminaceae and solanaceae maintain viability for months. Correct. In few species, the seed lose viability within few months. Seeds of a large number of species. How long do the seed remains alive after they are dispersed? This period again varies greatly. In a few species, the seed lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. How long do the seeds remain alive after they are dispersed? The period again varies greatly. In a few species, the seed lose viability within a few months. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. How long do the seeds remain alive after they are dispersed? This period again varies greatly. In few species, the seed lose viability within the few months. Seed of a large number of species live for several years. Seeds of a Some seed can remain alive for hundreds of years. There are several records of very old yet viable seed. The oldest is that of a lupine, Lupinus arcticus, excavated from Arctic tundra. Arctic tundra. The oldest is that of a lupine, Lupinus arcticus, excavated from Arctic tundra. Lupine, oldest. Okay, lupine. Lupine, I don't know. Lupinus arcticus, Arctic tundra. The seed germinated and flowered after the estimated record of 10,000 years of dormancy. Flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of the date palm. A recent record of 2000-year-old viable seed is of the date palm. Phoenix dactylifera, discovered during the archaeological excavation at King's Herod's Palace near the Dead Sea. King Herod's Palace, Dead Sea. Phoenix dactyliferus, 2000-year-old date palm. Okay, palm. Phoenix dactyliferus. Phoenix dactylifera. Phoenix dactylifera. Phoenix dactylifera, discovered during the archaeological excavation of King Herod's Palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following question: how many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? And so on. Can you think of some plants in which fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar is the case in fruit of some parasitic species such as Orobanchian striga. Similar is the case of fruit of some parasitic species such as Orobanchian striga. Very large number of seed. Orchid fruit. Okay. Orchid fruit. Very large number of seed. Similar is the case in fruit of some parasitic species such as Orobanche and striga. Parasitic species, okay. Striga. Have you ever seen a seed of ficus? How large is the tree of ficus developed from that tiny seed? How many billions of seed does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the years? Ficus, right? Wait a second. Ficus, F-A-C-U-S, ficus. So this one is, that is ficus seed. I feel it's so tiny. Ficus versus fig. Ficus is the botanical genus name for a larger group of plants, many of which are commonly known as fig or fig tree. Okay, ficus. Give me the fig tree. Okay. Orobanche and striga. Orobanche and striga. Broom rape. Which with? Stick. Are parasite plant in Orobanche family, okay? And their fruits are notable for producing a large number of very small seed, okay? Apomixes and polyembryony. ठीक है तो है. Apomixes and polyembryony. Although seeds in general are the product of fertilization, a few flowering plants such as some species of Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization called apomixes. Okay, production of seed. Okay, apomixes का seed produce हुन्छ but without fertilization. Produce seed without fertilization. Produce seed. Okay, they do produce seed but without fertilization and are called apomixes. And they are called apomixes. Although seeds in general are the product of fertilization, a few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism. Asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization. Called apomixes. Seed in general are the product of fertilization of few flowering plants such as some species of Asteraceae and grasses. Seeds in general are the product of fertilization. Although seed in general are the product of fertilization of few flowering plants such as some species of Asteraceae and grasses. Asteraceae and grasses have evolved a special mechanism to produce seed without fertilization called apomixes. What is fruit production without fertilization called? Fruit production without fertilization. Parthenocarpic, isn't it? Fruit production. You are right. They are called parthenocarpic fruit. Fruit production without fertilization. Parthenocarpic. Thus, apomixy is asexual reproduction. Apomixes is asexual. Apomixes is a form of asexual reproduction that mimics sexual reproduction. But it is asexual reproduction, okay? Because formation of seed without fertilization, asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, okay, there are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division. The diploid egg cell is formed without reduction division. And develop into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellular cells surrounding the embryo sac starts dividing, protrude into the embryo sac and develop into embryos. Okay, nucellular cells से भी हो सकता है. Protrude into embryo sac and develops into embryo. As in many citrus and mango. Okay, the one is from diploid egg cell. Without reduction division and develop into embryo without fertilization. Diploid egg cell is formed without reduction division and develop into embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellular cells surrounding the embryo sac starts dividing, protrude into embryo sac and develop into the embryos. In such species, each ovule contain each ovule contains many embryos. Occurrence of more than one embryo is rare. In a seed is referred to as polyembryony. Develops into embryos. Okay. In such species, each ovule contains many embryos. citrus and mango varieties. Some of the nucellars surrounding the embryo sac starts dividing, protrude into the embryo sac, and develop into the embryos. More often, as in many citrus and mango varieties, some of the nucellars cells surrounding the embryo sac start dividing. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac starts dividing. Develops into the embryo without fertilization. In some species, the diploid egg cell is formed without reduction division and develops into the embryo without fertilization. More often, in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac starts dividing, protrude into the embryo sac, and develop into the embryos. In such a species, each ovule contains many embryos. In such a species, each ovule contains many embryos. Occurrence of more than one embryo is in a seed. In such a species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Citrus, mango, and opuntia. Take out some seed of orange and squeeze them. Observe the many embryos of different sizes and shape from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryo? Can they be called clones? Hybrid variety of several of our food and vegetable crop are being extensively cultivated. Cultivation of hybrids has tremendously increased productivity. One of the problems of hybrid is that hybrid seeds have to be produced every year. If the seed collected from hybrids are sown, the plants in the progeny will segregate and do not maintain hybrid characters. Production of hybrid seed is costly, and hence the cost of hybrid seeds becomes too expensive for the farmers. If the hybrids are made into Apomixes, there is no segregation of character. In the hybrid progeny, then the farmer can keep on using the hybrid seed to raise new crop. Year after year, and he does not have to buy hybrid seed every year. Because of the importance of Apomixes in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of Apomixes and to transfer apomictic genes to hybrid varieties. Polyembryony, citrus, opuntia, mango, citrus, seeds without fertilization. मेरा चैप्टर कंप्लीट हो गया टाइम से पहले वैसे, बट 6:30 हुआ भी नहीं है। वाह। कितनी खुशी की बात। अब मैं पढ़ूंगी एवोल्यूशन पढ़ूंगी शायद। एवोल्यूशन ही पढ़ूंगी। Banana example of parthenocarpy, honeybee example of parthenogenesis. आई थिंक हनीबी का तो मुझे एक ही पता है, ये हनीबी का जो मेल होता है ना वो माइटोटिक डिवीजन करता है, इफ आई एम नॉट रॉन्ग। ये नया एनसीईआरटी में दिया था, मैंने कल ही पढ़ा था शायद। लेट मी चेक ला 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 ला। ब्रेक टाइम हो गया। ओके हम लोगों ने 90 मिनट से ज़्यादा पढ़ा, 100 मिनट जितना पढ़ा। ब्रेक 10 मिनट। आई हर बार ये नोटिफिकेशन आता रहता है। मैंने सही बोला। हैप्लोइड सेल्स डिवाइड बाय माइटोसिस, फॉर एग्जांपल मेल हनी बी। एनिमल में भी माइटोसिस, हैप्लोइड सेल डिवाइड बाय माइटोसिस, फॉर एग्जांपल मेल हनी बी। नोवा इज देयर। ओके किस किस ने ऑनेस्टली बोलो ये स्लॉट कंप्लीट किया? नोवा ने कंप्लीट किया था क्या? मैं ब्रेक को पिन कर देती हूँ ना। ये लो पिन कर दिया। पढ़ने के लिए तो कोई नहीं आते। ब्रेक एंड होने से पहले मुझे जाके खाना खा लेना चाहिए ना। You did complete it, okay. Shri, are you there? I think he left, but I'm not sure. Shri, are you there? Someone is saying he can't say his real name. ऐसा क्या खास है नाम में? नाम ही तो बताना है। कोई चोरी करके बैठे हो क्या? We'll have some coffee. Okay, I'll have rice. Okay, I'll come back before the break gets over.